Yes, thank you. So good morning, everyone. Today I'll be presenting some of my master's work on the post-harvest mold of pome fruit stems and calyx sepals, looking into the investigation into the incidence, cause, and management under the supervision of Dr. Shea Lennox and Dr. Julia Mites-Hopkins. So what is calyx and stem mold? This is a superficial mold. So what is calyx and stem mold? This is a superficial mold that is restricted to the fruit stems and calyx sepals of fruit coming out of controlled atmospheric conditions. These molds appear as a white to grayish fluffy mycelial growth seen on the stem or on the calyx. Literature first reported these molds as a cosmetic mold occurring on pears in Argentina. From the study, two prominent pathogens were identified, that being characteristics of Alternaria and Cladosporium species. These molds were first identified on pears coming out of long-term storage before packaging occurs, but more recently, packhouses have identified these molds on apples as well. Therefore, these molds lower the fruit quality and contain a very negative reputation with industry, further affecting future sales. These molds have major cost implications with industry. Therefore, the aim of my study was then to provide the pome fruit industry with effective and appropriate management solutions to this problem on the stems as well as on the calyx. The objectives I'll be focusing on in this presentation is firstly then to look at the identification of the major fungi present, secondly to do 3 in vitro tests, look into the sensitivity of these isolated fungal groups to registered post-harvest fungicides, Fludioxonil and pyrimethanol. So how this was done for my identification, symptomatic fruit material was isolated on PDA plus media, where pure cultures were obtained and culture plates were grouped according to how they look morphologically using the microscope. For molecular identification, mycelia was harvested and prepped for DNA extractions, where PCR amplifications was done, looking at various gene areas for the specific genera groups. From this, samples were sent to the sequencing facility at the Stellenbosch University, and then upon arrival, these sequences were then blasted on the NCBI database for further phylogeny analysis. So from our morphological identification results, 3 main groups were identified, that being characteristics of Alternaria species, which was a bulk of our isolates, approximately 78%, and then in lower frequencies, we had approximately 10% of our isolates were characteristics of Cladosporium species, and then Epicoccum species. It's important to know that our molecular identification to species level is currently underway. For our sensitivity now to Fludioxonil, we grew out 22 isolates, calyx or stem mold from apple and 6 from pear. Fungicide amended media was prepped, whereby PDA media was amended with Fludioxonil at various concentrations, starting at our lowest 0.01 and our highest 1 milligrams per liter. Fungal growth or mycelial plug containing the fungal growth was then inoculated onto these plates and incubated for 5 days in the dark at room temperature. From this, mycelial growth can be measured using a digital caliper, and the data statistically analyzed to determine the EC50 and the EC95, which is then the effective fungicide concentration inhibiting 50 and 90% of the fungal population. So this is just some results. So the graphs here depict the average EC50 values across the 3 main groups. For Alternaria, our EC50 range was 0.03 to 1 milligrams per liter, with one isolate A8 being less sensitive to Fludioxonil. Now looking at Cladosporium, our average EC50 range was 0.06 to 1 milligrams per liter, with standout isolates being less sensitive to Fludioxonil, C1, C4, and C6. Now looking at the Epicoccum group, we saw that the average EC50 range was 0.03 to 0.64 milligrams per liter, with E8 being less sensitive to Fludioxonil. So what this all means is that from this study, we saw that there were 3 main groups identified, that being Alternaria, Cladosporium, and Epicoccum. It's important to note that there were other fungal groups identified, but in lower frequencies. From this coincides with literature whereby Alternaria and Cladosporium species were found causing calyx and stem mold on pears. For the sensitivity tests, we saw that the Alternaria and the Epicoccum groups were more sensitive to Fludioxonil than Cladosporium species. Also within our study, we saw that it possibly suggested that these pathogens could be present already in the orchard and under favorable conditions allow them to proliferate, appearing as this white fluffy mold either on the stem or on the calyx. Through our sensitivity results, this gave us good insight into current management strategies, and also what we tested within the study, we looked at alternative management strategies by the use of various packaging types, if this had any influence in mold development. Thank you.